Hi, Dr. Suji, it's BT, and we're going to be talking about our gut microbiome lab. So, microbiome is microorganisms living in a particular environment. The gut microbiome are the microorganisms found in the gut. And the relationship between gut microbiome and physiological conditions. One example is scientists replaced gut microbiomes of obese rats with normal rats, and it made the obese rats lose weight. The second example was fecal microbiota transplant was used to help with Clostridium difficile. This helped prevent liver failure. And our question was, how do the diets of various reptiles differ? And our hypothesis was the bearded dragon and gecko's diet would be very similar, while the corn snakes would be different. So for materials and methods, we tested a corn snake, a gecko, and a bearded dragon. They were all predators and reptiles. The corn snake eats much larger prey, while the bearded dragon and gecko eat insects. To purify the DNA, we used the cryogen kit, and we followed the lab manual exactly. To find the concentration, we used the Zymo kit, and to find the quantification of the DNA, we used a nano-drop spectrophotometer. Through this, we found the purity of the sample and determined if there was contamination. The Amplicon PCR we used to amplify the V3 and V4 regions of the 16S ribosomal RNA gene. And for the gel electrophoresis, we did this to separate DNA fragments of different sizes. The PCR cleanup was used to remove old primers and PCR buffer. And for our results, you can see that for this sample, we found 5 different types of bacteria. And for this one, we found 3. And for this one, we found 5 as well. And for discussion, the extraction was unsuccessful because 2 of 3 of our samples did not work. A high enough concentration means that there's a good amount of DNA. The higher the concentration, the higher amount of DNA. Most of our results did not work well. We only got a band for number 4. And our negative control also worked because it didn't show up. And there must have been cross-contamination due to the failure of our samples. And yeah, the negative control should have been 0, which it was. And yeah.